first thing I want to do is just have any of you or your children participated in a clinical trial for a drug for Alagille syndrome? Just to get a, okay, so you guys, yes. And you, okay. And you're both in the Shire study, yes? Okay. And you're in that right now.
Well, technically, I think it's the only.
It's the only drug, actually, yes, it's the only drug study that's going on ever. So, you know, that's the only gig in town.
Yeah. I was saying to my husband last night, I said, I think it's a scary fact that I could possibly be the world expert in Alagille syndrome clinical trials. How scary is that? But that's going to change.
Very, it's going to change.
It's really going to change because there's a lot more interest from companies now, and research institutions, and research yields new drug ideas. So I think that this is like the beginning. This is the tip of the iceberg of the future.
Mm-hmm. Yep. And then we just talked about basic, you know, versus or basic to clinical, and you know, what happens where you go from drug discovery or chemicals and molecules in a laboratory to the clinic eventually. And I guess I'd like for Kira to just give us you know, the drug discovery process is a long process. You start out with thousands of compounds, and you might end up with one if you're lucky, you know, that will make it through.
So that's the traditional view of drug development. So my husband's a project manager, and he's in the engineering world, and I remember the first time I developed my first, from discovery through to theoretical approval timeline on a Gantt chart using Microsoft Project, which is the same software he was using for his engineering projects. And his we have a fan. You know, his timeline was, 15 months. Mine was 17 years. You know, I mean, that's the difference. To go from a concept, a target, to do either biological intervention or small molecule medicinal chemistry, to narrow it down to a scaffold and do some more medicinal chemistry or engineering or tweaking about that, and then you go into lead candidate selection and preclinical toxicology, pharmacology. Then, you know, maybe you're ready to start thinking about your clinical trials to get all the way through to approval. That is typically a 15-year process. Now, it's also, I mean, there's lots of numbers that are thrown about in the drug development industry about it costs $1 billion to develop a drug. And I think that those numbers can be misleading, and they're also politically charged numbers that are used for certain lobbying efforts. But in reality, if you take from the beginning of research and you add up all of the dollars that are spent, and you look at the end of the pipeline, how many drugs get approved, and you spread those costs across all those efforts, it probably does cost $1 billion. Now, in rare disease, no company is going to spend $1 billion or think about that type of a program, because the commercial opportunity at the end of the day is not the same as it is for diabetes or cardiovascular health or cholesterol reduction, where you've got hundreds, tens of millions of patients. You've got a couple thousand patients at best in some cases, a couple of hundred patients in some cases. So I think that in drug development in rare disease is an entirely different beast. And FDA acknowledges that, and they think about it differently. Doesn't mean you can take massive shortcuts, but usually I think what happens, and what we did at Lumina, was we had a drug that had a lot of data around it, that had been through all of that research and discovery and preclinical and phase 1 and phase 2 for in a different disease. And we licensed that drug, and we kind of turned it on its head and took it in a different direction. But we were able to gain a lot of leverage for all of that data that came with that drug. So I think that in rare disease, you see a lot of somewhat more creative thinking about drug development. Doesn't mean there's no de novo discovery research to come up with a new strategy. There is that too. But even in that case, I think it's more targeted, more specific, and you try, particularly in the clinical development phase, to be a lot, you get a lot more out of your trials because you're not going to have as many patients. You can't rely on the numbers, you have to rely on the details.
So for those, I'm assuming that most people in here are not familiar with, taking a drug from preclinical through to phase 1, 2, 3.
So FDA has some requirements that they will not budge from. In order to initiate a clinical trial, you have to have toxicology studies conducted in 2 species, unless you have a valid argument as to why there's really only one relevant species. And it's rare that anything gets into the clinic without having that. So usually it's a rodent species and a non-rodent species. So rats and dogs or rats and monkeys, typically. And you need to have enough what's called exposure, so days on drug in those animal species to cover the days on drug that you're planning to dose in the clinic. So usually you start off with a 7, 14, or 28-day preclinical study to cover your phase 1 studies, which typically don't go over 14 days. And then while you're doing that study, you start another study that's a 3-month study so that you can dose for 3 months in the clinic. And then you do a chronic toxicology study, which is 6 or 9 months depending on the species, so you can dose long-term in humans. So it's sort of a back and forth seesaw balance of the data you have preclinically to support the data you want to generate clinically. And that's all about safety. That's all about ensuring safety. And it's an imperfect model, but it's the model we have, and I think it's the best that we're going to have unless you can really engineer cell-based systems that truly replicate what happens in the human body, and we're a long ways away from that.
So investigational drug. New drug.
So you file what's called an IND, an investigational new drug, which is a bunch of paper, or electronic now, thankfully, that you can submit to the agency. And there are 3 primary sections to that. You have your preclinical section, which details the rationale, why you think this drug is going to work, what evidence you have to support, so what's called the pharmacology data. And then you have the toxicology data as to the safety profile of the drug. And there's lots of other bits to that you can look at: genotoxicology, cardiovascular toxicology. So is it likely to impact the heart? Not that that means you can't take it forward, but it means you should be looking for something in your clinical trial. So all of your preclinical information dictates what you think is going to happen clinically and what you should be looking out for clinically. Now, if you have a really bad tox signal, it may be challenging to go into healthy volunteers. But if you have a really, really sick patient population that has a short lifespan, then you might be able to get permission to go into patients straight away. So then you have, that's section 1. Well, actually, it's different modules to module 3 and part 4. So then you have what's called your CMC, your chemistry, manufacturing, control. How did you make the drug? How do you ensure the quality? What are the specifications? So that the engineers in the room know all about that. That's a very formulaic section in the IND. And then you have your clinical section. So when you're first going, your very first IND essentially has no clinical section because you've never been in humans before. As you evolve your program and do clinical trials and go back to do more clinical trials, then that section evolves and grows. But the clinical section in the first IND has a protocol. So what are you going to do? What are you asking? You're asking the FDA for permission to do something. So that's why you file an IND. You're typically asking them for permission to do a study. It might be a first-in-human study. It might not be a first-in-human study. It really depends on the program. But you're asking them to say yes to this protocol. And that's the third sort of section that goes into an IND. And typically, these can be it depends. A very first-in-human IND could be several thousand pages. Our IND for Lumina, because we had all this legacy information, The electronic file was 40 gigabytes.
Hmm.
Yeah. So tens and tens of thousands of pages
Right.
that the FDA reads.
Believe it or not. Valley of Death.
Actually, I'm joking. The FDA is really good. They really do take their jobs very seriously, and they do pay attention to things. Now, there are summary sections that are written to individual study reports, but you would be surprised at the level of detail that they will go through in this review. And once you submit an IND, they have 30 days to tell you whether that study is approved or not. So, I personally would not like to be working at the FDA.
Neither would I. So, Valley of Death, bridging the gap from molecule to, yeah.
Like many things in life, early stage drug development is not for the faint of heart. I think it is an exercise in persistence and an exercise in really stringent scientific thought rationale process. So in some ways, to the researchers in the room, you can design an experiment that's going to get you an answer that you're going to like, or you can design the really hard killer experiment it will really decide if you have something. And I think the more diligent we can be to design the killer experiments, the better for the community and overall. And again, like many things in life, timing, being in the right place at the right time counts for a lot. So having a dedicated champion for a program is often what makes or breaks whether something actually makes it into the clinic and gets that way, it gets across.
Mm-hmm.
Anybody have any comments or questions? Feel free to
Yeah.
comment, because I think that we should make this as interactive as possible.
Mm-hmm. All right. So, what might be, I guess I want to, at each stage in the process, so we've got preclinical up to the IND. Yeah. Is there a role for ALGS patients, ALGS caregivers in that early stage?
Yes, there absolutely is. And I think that animals tell us a lot. Human cells tell us a bit more, and human subjects tell us the most. So you heard Henry this morning talk about collecting blood and skin samples. Sometimes those tools are invaluable to researchers. So donating blood or donating skin samples or tissue or doing the hearing tests, things like that, they can really help refine your clinical thinking about what you might expect in a study. So I know that the O'Shea's participated in our qualitative research when we were assessing symptoms and severity of symptoms and burden of symptoms and how you think about symptoms, because there's clinical endpoints. So in Alagille, there's itching, there's GGT, alkaline phosphatase, all delivered biochemistry. There's cholesterol, xanthomas, growth, fibrosis, cardiac functioning. There are so many clinical symptoms, but there's also what are called patient-reported outcomes, so quality of life. Itching is a patient-reported outcome, so that somebody saying my itching is better, it's worse, it's the same, and those are tools that are, again, imperfect, particularly when you're trying to figure out how itchy a 2-year-old is, or younger, and at what point can a child really reliably consistently report their itching. And itching is so subjective. So, if there's an opportunity to share your point of view on your experience or of your child's symptoms, then that really helps us as drug developers think about, okay, what is my endpoint? How do I measure it? Well, how am I going to roll that out? And how do I do it in a way that's going to be of the highest scientific integrity? So those are the times when you can really influence how many options there are in the clinic for this condition.
Well, and I would add to that too, the listening sessions that we had, just sharing that experience and what it's like for you and for your child really helps inform researchers' work. And it helps them think about what might be new endpoints, what might be, new targets for drugs that they're developing. So.
Right, and I mean, the FDA is trying to advance new programs that really are tailored towards rare disease. One is called the Breakthrough Designation Program, and that is a designation that's given to a drug candidate, because it's not really a drug until it's approved, a drug candidate that says, check, this drug shows clinical evidence, so not preclinical, it has to be clinical evidence of a significant effect in a high unmet need patient condition. And then you can have a more accelerated path to approval, more interactions with the agency. It's interesting when you think about, if you haven't lived how you interact with the FDA, you would have no idea how broken the system truly is. So if I want to talk to the FDA, if I have a question for the FDA, I submit a meeting request, and there's type A, type B, and type C. Depending on that request, I will hear in 1 month or 2 months when my meeting is. And it could be 60 days later, 75 days later. If it's a real emergency, it could be 30 days. And then I send them a briefing document, which takes an inordinate amount of time to prepare. And they read that, and then we have our meeting. And then 30 days later, we get the minutes from the meeting. So to get an answer from FDA, you're talking about a 6-month investment of time, and in some ways, 6 months of wasted time. I think the Europeans do it a lot better. They have a lot more, certain countries have a lot more sort of, it's almost like a guidance. You can call them up. You have a bit more flexible interaction. And I didn't realize until my head of regulatory told me, when the FDA tells a company something, that's like policy. It's like legislation. So they have to be very careful about the way in which they communicate things, make sure it's consistent with all the previous guidance that they've given other companies. And the scale of the US and the numbers of drugs and the numbers of companies, it's exceedingly complicated. There's no sort of FDA get out of jail, phone a friend. Hey, I'm thinking about this. What do you think? Yeah, no. I mean, we don't want binding information. We don't want to be able to say, well, you said this in these minutes, and therefore you have to do this. And I actually think it's something we didn't talk about in research, the research session. Alagille has a really hot connection at FDA, and I don't know who knows about this, but there was a former fellow of David Piccoli, whose name is Andrew Mulberg. He's the Deputy Division Director of the GI Division at FDA. And Alagille falls under the GI and Inborn Errors of Metabolism Division. So there's an advocate within FDA. And I've seen how that can help companies come up with the best plan for Alagille syndrome. And who knows how long he'll be there. So,
Yeah, it's been a few years. And actually, the study, the pancreatic insufficiency study that I talked about, that was Andrew's study when he was at CHOP.
Yeah. So, he's a huge asset for Alagille syndrome at FDA. And hopefully he'll stay there for a very long time. Typically, the longer they stay, the longer they're going to stay. So, I think that there's an opportunity for researchers to have better insight and dialogue because Andrew's there and because he understands the condition. He understands the disease. And when you have Cindy on the phone saying, you treated my daughter, the patient voice is loud and clear.
Yeah, Andrew and I go back a few years, yeah. So clinical trials. So we've got the IND.
Right,
and we're heading into clinical trials.
So just to understand the nuts and bolts of getting a clinical trial open. So first of all, you have to file your IND, and the FDA has to say yes. And then you have your sites. So let's take CHOP as a site, for example, Children's Hospital of Philadelphia. they have their own review board called their IRB, Institutional Review Board. So you submit a package of information with the protocol, the informed consent, a document that's called the investigator's brochure, which is basically the physician's cheat sheet to everything that's in the IND in summarized version. And that all goes, and a few other things, go to the Institutional Review Board. So FDA is saying, yes, you can do the study. They're not saying anything about where in the US you can do it, and they only have, jurisdiction over the US. The Institutional Review Board is saying, yes or no, you can do the study in this hospital. And in the meantime, you're negotiating budgets and contracts with the hospital because, this is a big part of their business. And, sometimes it just does come down to budgets and contracts. So once the FDA says yes and the IRB says yes, then the company has an obligation to go and do what's called a site initiation visit. So you go and you make sure that the staff understand the protocol. They have all of the tools, the electronic diaries, the lab kits, the manuals that they need to properly execute the study. And once you've had the FDA saying yes and the IRB saying yes and the site initiation visit complete with all of your check marked and checklist done, then that site can enroll their first patient.
And if you have more than one site, you have
to do that
multiple
every
IRBs,
time.
and you do that
Yes.
at every site.
There is a thing called a central IRB where some institutions defer their IRB privilege to a central IRB. So say there's there's a number of them. Western IRB is a big one, and these three hospitals say we agree we accept Western IRB. So Western IRB says yes, then these three hospitals are checked. But these two have their own IRBs, so it's a very complicated process. Now, if you're doing a global study, you have to get the FDA and all the IRBs to say yes. If you're doing a study and you have to get a study in the UK, you have to get the MHRA to say yes. You have to get they have a central ethics, or IRB, for the whole country. But then you also have an additional review process at the hospital. And every country is different. Every site sometimes can be different. So a lot of what we internally do is paperwork and shuffling things from one to another and answering questions. And every physician and hospital and regulatory agency have to do what's in the best interest of their patients and feel comfortable with the risk benefit. It always comes down to risk benefit. So the safety profile of the drug is really important. And risk benefit changes. Again, if your child's perfectly healthy but they have a recurrent earache, your risk benefit profile is going to be very different to your child who hasn't slept through the night in 12 years because they're itching themselves and tearing their skin off, which has a very different risk benefit to, we have a neurodegenerative disease and I know that my child's not going to see his teenage years. So, it depends, and you really have to have the specific context of the patient population and the drug in terms of how can you inform a parent to say, this is what you should expect. These are the things that could happen. These are the risks. And, when you've read informed consent documents, there's lots of risks. And they don't talk a lot about the benefit because we don't know yet for sure there's theoretical benefit. Yeah. And clinical trials might be 1 day, 1 week, 1 month, 1 year, 10 years. It all depends on what the study is designed to address. And there's no one size fits all.
Phase 1,
phase
phase
1.
2, phase 3. Let's start with phase 1.
Phase 1. And I'm going to talk generalities because there are exceptions to everything. In most cases, for small molecule drugs, so for a pill, or sometimes you can administer small molecules lots of different ways, but just let's use the example of a tablet or a liquid suspension for oral administration. Phase 1 is what happens in healthy adults. That's where you start. You start with healthy adults, and then if your trial or your drug is really targeted towards children, then the FDA or whichever regulatory body, if you have the right preclinical package, there's a juvenile toxicology package that you might have to do before you can dose children, you can start working your way down in age to younger and younger children, because the body matures as these children grow. The GI tract is different, the brain is different, things are evolving over time. So the risk benefit, again, is highest in the least developed babies. So you really have to understand what happens with the drug. So how safe is it? What are the side effects? What is the PK? Now, what's PK? PK is an abbreviation for pharmacokinetics. So where does the drug, how much drug gets into the blood system? How high is the highest concentration of blood in the blood system? How long does it stay? So it's called a half-life. So there's a parameter that says, how long does it take for half of the drug to get out of the system? Then there's, how's the drug metabolized? Where does it go? So these are all the things that you research in phase 1. So the Lamina drug is an interesting one because it has no PK. It's not, it doesn't get into the blood system. So it's an orally administered drug that stays in the GI tract and is excreted in the poop. Here we go again. It's always all about poop. Essentially in the same form as you swallowed it in. That's very atypical for drugs. Usually they get metabolized by the liver. And if you're treating a patient with liver disease and your drug is metabolized by the liver, that gives you a little pause for thought as to is that going to cause any problems? Mm-hmm. Do the metabolites have activity? Do they go somewhere else? How long do they stay in the body? What's the peak concentration of those in the blood? So these are all of the sort of basic characteristics that you work out in phase 1.
So in a phase 1 trial of Alagille syndrome, how many patients would you have?
So I think, depending, again, everything is, I went to business school and I learned that the answer to every single question is it depends.
It depends.
But if you have a small molecule, I don't think there's there's an average phase 1, but it wouldn't be in Alagille children, mm-hmm. for the most part. So it would be in adults, and I'm doing a phase 1 right now, and it typically a phase 1 has what's called a single ascending dose phase, where you start at a low dose, single dose, and then you go to a higher single dose, and then a higher single dose, and then a higher single dose, and you're really dosing to find out how high can I go? How high before I start seeing some dose-limiting toxicities? And then you start the multiple dose, then you go back down a dose and you do multiple doses, and then you go up to a slightly higher dose and do multiple doses, and et cetera. So our phase 1 that's ongoing right now will have 100 subjects in it. Mm-hmm. Each cohort, which is a group of patients who are all undergoing the same regimen, not necessarily the same drug or placebo, but the same duration of treatment, has 8 subjects, 6 on drug, 2 on placebo. So that's not an atypical phase 1 mm-hmm. design.
Okay. So
phase 1 to phase 2.
Phase 1 to phase 2.
So once you know the profile of your drug, and once you know all about the PK and where it goes, and how to dose it, and how high you can dose, and if you know that you need to have a certain level of drug on board because that's what your animal models say that you need to have the best effect, and if you have to dose that once a day or twice a day based on your phase 1 work, then you have a dosing regimen that you can test. Or maybe you have likely you have multiple dosing regimens that you can test. And this is when you move into patients. And this is again a place where rare disease and the rest of the more common diseases really diverge, because you don't have thousands of patients to test 5 different dosing regimens on. So there are multiple phase 2 designs, and a lot of things die in phase 1. A lot of drugs die in phase 1 because the safety is not acceptable to go forward with. So that's the first thing about phase 1. If you get out of phase 1, it's good. Mm-hmm. It's really good. And now I think, depending on the drug and depending on the mechanism, you may be able to get some readout of efficacy in phase 1. So again, I don't mean to be talking a lot about Lamina and the Lamina drug, but mm-hmm. I can give you some examples. So healthy people have bile acids, and the Lamina drug theoretically should lower bile acids. So we can see in phase 1 if the drug is doing what it was designed to do, and it did. So we knew that the drug was hitting the target, it was having the desired effect, and then we move forward. Now, I say that as if we did phase 1 intending it to be for Alagille, but we didn't. We didn't do phase 1 at all, so other people did phase 1. But you can tell from the data. You can sometimes get a read. Now, in my current company, I am developing drugs for invasive fungal infections. So healthy people don't have fungus and fungal infections. So we're not going to get any efficacy readout from our phase 1s at all, because there's just not that clinically relevant situation. So it depends on the drug and the target, whether you can get any hint that your drug is doing what you think it's going to do when you get into patients. So anyway, with all of that forwards. So phase 2, typically you try to test multiple doses. And in rare diseases, you try to get a bit creative here, because you don't want to dose too low and miss an effect. You don't want to dose too high and patients leave the study because of safety or tolerability. You don't want to hurt anybody. Mm-hmm. So we had a number of different designs that we did in parallel. The U.S. study has started off with 3, now it has 4 cohorts or 4 doses. You have placebo, you have 70 mg per kg, you have, actually not 70, that's the adult dose. Yeah, 70, yeah, let's call it dose A, I can't remember. B and C going up low, medium, and high doses. So patients come into the study, they get randomized into one of those 4, and then they stay in that group. Mm-hmm. What we did in the UK study was we started the first cohort with the middle dose, and then after a while, when we felt we had enough information on safety, we decided we had a predefined strategy. If the safety meets these criteria, then we will go to a lower dose for the second cohort. But if the safety does not meet these criteria, we go to a higher dose for the second cohort. So we have the ability to have a protocol that allows us to evaluate multiple doses without dosing too low and not seeing anything, or dosing too high and not having a worse safety profile. So that's what we did in the UK study. In the global study, we had, which is a design that is actually gaining a lot of traction in rare disease. It's called a randomized withdrawal. So the first 18 weeks, everybody was on drug. And then at week 18, there was a randomized withdrawal. So patients were randomly 1-to-1. When I say 1-to-1, I mean one patient was withdrawn from drug, one patient wasn't, versus a 2-to-1 where maybe 2 patients would be withdrawn and one would not, or vice versa. So it's just a ratio. So randomized 1-to-1 withdrawal from drug for 4 weeks, and then everybody put back on drug to 48 weeks. So what that and each design tells you something different. What that design tells you is, what does the drug do and what happens when you take the drug away? Which is an important question that FDA always worries about, because if when you take the drug away something bad happens and you need to taper, then that's something you need to know, and you can all about guiding the physician as to how they dose and use the drug. Mm-hmm. So, we also had open label studies, which is, they're not randomized, there's no placebo, everybody gets drug. So it really depends on what question you want to answer in your clinical trial. Like, typically it's, does this drug work compared to placebo? Mm-hmm. And in which case you have a placebo group. And that's, I'm sure that's tough for parents, knowing that there's a risk my child's going to get placebo. But that's actually one of the, that's the most guaranteed way that if the drug shows a difference, that the drug's actually going to get approved. So in almost all cases, placebo-controlled randomized trials are required for approval, unless the clinical course of the condition is so well known, established, and so severe that in an open label setting, you can see a difference that cannot be influenced by subjective knowledge that you're on a drug. So if children, have liver failure by the time they're 5 and they're on drug, and they don't have, none of them on drug have liver failure, and then you have a very clear sort of black and white scenario, but those are actually the exception.
And the Shire study is phase 2.
Yes.
Okay. Phase 3.
No, it's okay. So
Moving
the Shire
along.
studies are phase 2.
Yes.
But they were designed to be phase 2s upon which you could approve.
Okay.
So that's another rare disease exception.
Huh.
So FDA doesn't say you need to do phase 1, phase 2, and phase 3. FDA says you need to demonstrate safety and have either 1 or 2, typically 2 well-designed randomized controlled trials. That could be a phase 2 and a phase 3. It could be 2 phase 3s. It depends. So we designed the Lumina trials in a way with input from the agency that they could be submitted for approval. So in that sense, there would be no phase 3. Now, Shire, and I'm kind of speaking out of school a little bit here in terms of, we didn't know much about how this drug would behave in Alagille syndrome. Actually, we knew nothing. We had ideas, but they learned a lot, and maybe they have to go back and do another trial. Maybe they don't. I really don't know. But in the case where you have to do a phase 3, typically phase 3s are a repeat, slightly larger repeat of a successful phase 2. So you saw a signal in phase 2. You know how many patients you need to show this much separation between your placebo and your treatment, and you do that in phase 3. Usually you add a few more patients just to make sure that there's nothing weird, because the thing that really impacts clinical trials and clinical trial design is variability. So if your endpoint is, say it's cholesterol, and xanthoma is cholesterol. So your endpoint is, your cholesterol is 2,000, 2,000, 2,000, 2,000, 2,000, 2,000, then you don't really have a lot of variability within a patient. Yeah. FDA does not like recycling patients for phase 3. So if you have to do a phase 3, then typically you're looking for new patients. Now, there is a whole world opening up of what's called adaptive trial design, where you have patients and you enroll them into a phase 2, you do a pre-specified interim analysis, but they keep going. Maybe you add some more patients and that becomes your phase 3. So that you technically have the same patients, but it depends on your endpoint. Because if your endpoint is a change from baseline in something, and your patients in your phase 2 have had that change from baseline, they don't really help you in your phase 3 unless your endpoint for your phase 3 is baseline from enrollment to any point in the future. So it depends, but you typically, FDA does not like you to enroll patients in a phase 2 and they finish that study and then you bring them back for phase 3. But again, for rare disease, they may make the exception if whatever you're measuring has gone back to their previous baseline or has gotten worse again. And you may or may not want to enroll those patients depending on how effective your drug was. Like if your drug was truly disease modifying, you might not want those patients in your phase 3 because you've already conveyed a significant benefit through the drug that you might not see again by more drug. Right. But if you have a phase 3, so there's a designation called the Orphan drug designation, which is and there's an Office of Orphan Disease, the Office of Orphan Diseases?
Rare Disease, Office of Rare Diseases
Office of
Research.
Rare Diseases Research. Thank you. So if you have an orphan designation, and what you have to do is submit proof to the agency that there are not more than 200,000 patients in the U.S. with this disease. So, Alagille syndrome is a shoo-in. Then there is legislation that says if you have an orphan designation, you only need 1 randomized controlled trial. So that could be a phase 2, it could be a phase 3. It kind of depends on how it's designed.
DNA or NDA. Not DNA, NDA.
NDA.
New drug application.
So an NDA is a new drug application for a small molecule. If your treatment is an antibody or a protein, so what's called a large molecule, that's called a BLA, a biological license application. So, but essentially they're the same thing. It's where you're presenting to the FDA all of your preclinical data, which has continued to evolve since you filed your IND. It may include things like carcinogenicity studies, which are 2-year studies. It may include a variety of other things that help the agency. Ultimately, you know that piece of information that's inside Tylenol that nobody ever reads with the teeny tiny print? That's what's called your package insert. That is a highly every word on that page is negotiated to the nth degree, because it really is to do two things. It's to instruct the physician how to dose it and instruct the patient about the potential side effects. So everything that goes into your NDA can end up in that language. And what the FDA wants to say is, how do I write this section? Do you show me the information that allows me to write that section in that label, that package insert? So you have that huge preclinical section. You have a huge CMC section, which is the chemistry, manufacturing, control. Now you have an added burden. You have to show that you can reliably, reproducibly manufacture this drug at large enough scale to deliver your commercial need. So the last thing the FDA wants to do is approve a drug, and 6 months later you have a drug shortage because they didn't have a big enough scale of manufacturing ready to go. And then you have your clinical section, which is essentially broken into two pieces: the safety, the integrated summary of safety, and the integrated summary of efficacy. So all the side effects and the upside. And that's like millions and millions of pages.
We already talked about safety and effectiveness. When would you do a phase 4?
Phase 4s are after the drug is approved. So they're what they're called post-approval. Well, actually, there's two kinds of things. There's the phase 4s that the FDA gives you or mandates as a condition of approval. Those are called post-approval commitments. Or there's the things that the company might want to do in a phase 4 setting. But any trial that's done on an already approved drug is a phase 4. Even if it's a phase 1 type study, but it's on an approved drug, it's essentially a phase 4 study. So if, for example, Shire got SHP65 approved for Alagille, but was also studying it in other conditions, like maybe they wanted to try biliary atresia, then that would be a phase 4 study. But typically, the most important phase 4s are the ones that the FDA says, we're giving you approval to sell this drug. And that's really what the approval is all about. It's to market and sell the drug. You have to do another study in children under 12 months because you haven't studied those yet. Or you have to do a study for 10 years to show that this drug actually prevents liver fibrosis. Or you have to do a study in adult Alagille patients because you haven't done anything there yet. So it generally is a series of studies that bridge the gap from the clinical package. And with rare disease, there's often more gaps because the population is more well-defined and you don't have as many patient data to really base your NDA on.
Mm-hmm. So this is just a overview of the process where you might start out with thousands of molecules that you're looking at, and you might end up with
right. You have these things called compound libraries, or big companies have them, compound libraries, where a researcher comes up with a new target and you can screen tens of thousands of compounds to see if you get any hits, which is basically any sort of interaction, and then you kind of figure out, is it the right kind of activity to move forward. The numbers aren't quite as big for biologic drugs, and rare disease. The sort of quintessential, the first drugs that were ever approved for rare diseases were the enzyme replacement therapies, where the body is missing a specific enzyme. So a company goes and manufactures a large molecule enzyme, and you're replacing it. So this doesn't really apply to rare disease in my mind. This is generic drug development, but it doesn't apply so much to orphan drugs.
So the whole process for a drug, I know that with the Shire drug, that had been tested, and it was developed initially for a different application completely.
Yes.
And then it was picked up because it had been sort of shelved
Oh,
for a while.
it had
or was
been shelved, covered in dust,
Yeah.
forgotten about.
Uh-huh.
I mean, that drug was shelved in 2002.
Okay.
And we licensed it in 2012.
Right. So we're 4 years out from when you licensed it
Yes.
to
Yes, we are.
okay. And
Don't ask me how much longer.
I know. But really, but just as a comparison, to go from if you add the whole, if it hadn't already been developed and shelved and you picked it up off the shelf, if you add on, from initial discovery with a drug through
Right.
the
How
whole
long had it been worked
process,
on?
Yeah, we're looking
So-
at 10 years at least
Oh, at
to
least.
get through.
At least. So the prior companies and the drug sort of made it through a couple of mergers and died on the last merger, which was when Pfizer acquired it. So the drug was originally being developed to lower cholesterol. So you heard David yesterday talk about cholesterol as the mother of bile acids. And the theory was if you can force bile acids out of the body, you'll use up cholesterol to replace the bile acid pool and you'll lower cholesterol. And it worked. It just didn't work as well as statins. And the company, this drug started off at a company called GD Searle, which was acquired by Pharmacia, which was acquired by Pfizer. So in the early 2000s, Pfizer acquired a drug that was marginal at lowering cholesterol, and they had Lipitor. So it was a commercial argument. So the drug was shelved for commercial reasons. But at that point in time, they had conducted 12 clinical trials with over 1,400 patients. They'd probably been $150 million invested into the drug. And it's hard to think about a company investing $150 million into a drug for Alagille syndrome from the get-go. Yeah.
So before I go, I have some questions that I want to ask Kira, but first I want to see if you have questions that you want to ask Kira.
Oh, sorry. I didn't have the mic.
So I don't know if this is something you can talk about, but I know that seems like in the recent, year, 6 months, there's been a greatly increased interest in people looking at Alagille population and perhaps dealing with the JAG1. Can you give your any theories about kind of why the increased interest lately?
So I think the Lumina exit was a good one. And good exits create interest amongst investors to fund other companies to give them other good exits. I mean, I think that it's almost a self-fulfilling prophecy. So if companies are interested in a disease area, researchers become interested because you can develop new collaborations in a disease area. Not that that's the only reason researchers go into a specific disease area, but if companies are interested and they're from the Lumina perspective, the biology, the drug history, the disease population, it all just fit together very, very nicely, and it was a very financeable story. I mean, in drug development, you're telling a story. You hope to back it up with data, but you're really telling a story. And I think that exit, and to be honest, the initial data that came out from the UK study, I thought was very positive. Now, I'm not in Shire Communications, but it showed that when you can lower bile acids, you can improve symptoms and quality of life. And then there was other data in other conditions that was maybe a little less supportive, and the data that's been very supportive in other pediatric cholestatic conditions. So I think that it's just it goes from being a word nobody's ever heard of to, oh yeah, I remember that. Let's look at that.
Well, and I think I'd add to that as well that over the last few years, there used to be, when I started the Alliance, nobody had a clue what Alagille syndrome is, how it affects people, anything like that. No drug on the horizon anywhere, not even treatments. And we're stuck with the basic ursodiol. But I think over the course of the last 15, 20 years, there's been growing interest in not just our disease, but in Jagged 1, because it's implicated in a lot of other things. So there was a GWAS, a global
Genome-wide.
Yes, genome-wide something study, GWAS study, done that looked at osteoporosis in thousands of women. Jagged 1 is involved in osteoporosis in some way. There was just an article that came out last fall about golden retrievers in Brazil, that there's this one family of golden retriever dogs that exhibit signs of Duchenne muscular dystrophy, except for one dog and one of his offspring that do not. Well, when they did a genome, looked at their genome, they have a unusual amount of Jagged 1. And that is protecting them from having the signs of Duchenne muscular dystrophy. So there are other examples as well. And then the Notch signaling pathway is involved in many, many different disease states. And there's been growing interest in that. Well, we're also in the Notch signaling pathway, and one of the genes is Notch2. So I think it's just been a building, it's built over the years that now we're kind of sexy. Because our disease has Jagged, we've got Notch, we're interesting now. And, so there are not just drug treatments, but there are other things that are going on, just looking at the basic science, at the genetics, and a lot of people trying to figure out what's happening. And it's great. It's good for us.
And I think the regulatory environment continues to improve for rare diseases. So Orphan drug designation, breakthrough designation, all these things help companies justify spending dollars to develop drugs. And the Children's Network and associations like that have gotten people together to support this research. There's also some subtle things in the background. The current head of the Canadian Liver Association, Professional Association, is a pediatric hepatologist. The 2018 head of the American Association for the Study of Liver Disease is a pediatric hepatologist. So there are just some sort of environmental things I think are helping support pediatric cholestasis broadly, and rare disease broadly, that I think Alagille syndrome was well positioned for and have been able to sort of leverage.
I think another important point here is that the number of people who are suffering from rare diseases in the United States combined are more than those that are suffering from a combination of common diseases. But a lot of rare diseases are ultra rare, meaning that I work also on a different rare disease which so far has about 40 patients in the world. So among the rare diseases, Alagille is one of the common ones. Yeah. Because you can have
It's a bigger rare, yeah.
It's a bigger rare disease. Right? So that combined with is, I think, either the first or second inherited cause for childhood cholestasis. So even though it's rare, but it has some common features to it. And then, yes, connection to the Notch pathway, which is essentially in scientific circles, people sometimes dare each other to find a process in the body that is not regulated by Notch pathway. So we joke that if you find a process that is not regulated by Notch pathway, you can publish a big paper because it's almost non-existent. Every major process in animal development is regulated by Notch pathway. I think these are also reasons why Alagille is taking more attention now.
So there are three companies that were here, three pharmaceutical companies, and I think, Cindy, you mentioned one more that there was a lady that heard your speech. Can you give us a little bit of an overview what they are and what kind of things are they looking at? What drugs are they thinking of developing?
Sure. So the three companies are Shire, which acquired the Lumina assets, Albareo, which has a very similar product to the Lumina products. It's also an apical sodium bile acid transporter inhibitor. And then the third company is Sidon, and I can't disclose what they're up to, but it's a different strategy. It's very exciting, actually, but it's different. It's an entirely different thing. You know, I mean, other things that I think, clearly there are drugs that are used in Alagille syndrome, like ursodeoxycholic acid, but it's never, and it never will be studied. And rifampin never will be clinically trial studied. I think that there are other anti-fibrotic drugs that theoretically could be studied in Alagille syndrome, but the fibrosis in Alagille is not typically terribly progressive. So, I think that I haven't heard of others. I generally get a call from folks who are interested in Alagille syndrome drug development. And oh dear.
We do too.
Yeah. So have you, anybody else you're aware of? There's nobody else I'm aware of. No. So
No,
Sidon was the company that
Well,
heard you,
Sidon
right?
is, I think I can explain at least what kind of a company they are without saying anything other than, of what they're working on. But Sidon is it's what's called a rare disease accelerator. And so what they do is they look for promising research in rare disease. And when they find something, that might be going on in a lab, that might be, might be clinical, and don't know, but they look for promising research, and then they look for investors that want to invest in that research and move it forward more quickly, hopefully, than, and so that's who they are, and they have an interest in Alagille syndrome. So
I mean, they're actively doing work to come up with a drug candidate that would test their hypothesis.
Yep. More questions? Okay, so I have some questions. Oh, go ahead, yep.
As there is more drug development for Alagille, obviously there is more possibility of drug companies not behaving properly towards the patients and or at least not being extremely helpful. What recourse do we have? Like for example, this break in trials with Shire. What recourse as patients do we have? Is there anything that we can do to prevent this from happening?
Why don't you go first, and then I will give you my thoughts.
Well, I think there are there are several things that we can do. So one of them is, recently when, it was actually last fall, when we had things kind of blew up with Shire and the study that they're doing, and that the drug, several of our families were really concerned because they felt that, or they thought that the drug would not be available as an extension, and their children were having amazing results, even though the scientific data was not demonstrating the.
It was a different study.
Yeah, but yeah. So they were very concerned that this drug would not be available, and so one of the things that we did was we asked for testimonials from families. If you're participating in this study and you're seeing great results in your child, then tell us about it, write us about it. And we packaged all those together, and we sent them off to the head of the patient programs at Shire, the president of Shire, and said, we're concerned. We have families that are very concerned. And so we would ask that if you are thinking about, heaven forbid, dropping this drug and just closing the program, if you would read these and really think about how that is going to affect our families, how it's going to affect our patients. And that had an impact. I have since met with them a few times, and that really did have an impact. You know, another thing that I did was, and it goes back to kind of speaking and being out there, talking about the disease. I was invited a year ago, January of last year, to deliver the keynote address at Shire's Executive Leadership Summit in Boston. And got up there and just talked about, you know, myself as a parent of a child with Alagille syndrome, and then also presented my daughter's perspective. She was too sick to go. She was supposed to also be there. And I presented her perspective. And then I did that at the Leadership Summit. I did it at their Lexington campus later in the same day for researchers and staff. And it has an impact. You know, when researchers can see how if they can put a face on the disease, they can understand how this affects our families, then it gives meaning to what they're doing. There's also FDA, and there's the patient representative program that you can apply to. We can also, we may be setting up some discussions at FDA as well, because I've been talking with their Office of Rare Diseases, and to help them better understand our disease. You know, Kira has additional things.
You know, first of all, it's a horribly tough situation to be in, and there are a lot of factors that have influenced this particular development, and you know, the Shire is working with the Children's Network, and there's just a lot more pieces that have to fit together. But I think that everybody responds to pressure, and that pressure can be delivered through the Children's Network, through your physician, go through Ron Sokol, through NIDDK. I also, if I were in your shoes, I'd be talking to my congressman and you know, what's going on? You know, you never know who's connected to who. We're all people on the planet, and it's always worth making a stink about things. The squeaky wheel gets the oil. So it's not but in terms of, you know, legal obligations, there are none. There are none. Mm-hmm. No.
Yeah, and specifically with the Shire drug, you know, we're continuing, the Alliance is continuing to pressure them, to talk to them, check in every couple months and say, hey, how's it going? You know, do you need more information from us? You know, where's the study at? You know, what happens if, you know, you get the results back from the itch study and it's not looking so good? So what's the next step? Where are you going to go? And you know, we're doing everything that we can, you know, to advocate for the drug as well, because we've heard, you know, how much it affects our kids.
On your topic of getting out there and getting the word out, I just wanted to let everybody in the room know we're going to be having, I work for SBP, Sanford Burnham Prebys Medical Discovery Institute. We sponsor an annual Rare Disease Day symposium. I've personally organized 6, so I don't have a rare disease. I'm not part of a rare disease family, but I've been part of the community for a long time. And I can tell you from my experience that the basic researchers and translational researchers that I work with, most of them have never met a patient. And if you can get your family, your kids in front of their faces, it is inspirational to them in a way that no data, no pictures, nothing else can be. So if you're looking for a way to get involved and get your story out there, I would love for you to come and speak at our meeting. Get in front of these researchers.
The meeting is when?
Sorry, this is Rare Disease Day, February 24th, 2017. There's some flyers downstairs in the Crystal Ballroom if you want to pick one up.
And I think Lana has one right there.
Yeah, it's going to be at our La Jolla campus. Yeah, here in San Diego area. I'm the organizer. We have 2 keynote speakers. I'll refer to my notes real quick. One is our internal assistant professor, Duke Dong. The second is Holger Willenbring from the University of California in San Francisco, and all this information will be on our website soon. And then we have 2 keynote speakers who we confirmed so far, Nancy Spinner, who I believe most of you know very well, and also Dr. Raphael Copin from Cincinnati Children's Hospital Medical Center. We're looking at bringing in about 15 researchers overall. One is Dr. Jafar Najad, right there. And they will be in the 2 areas of Alagille, liver notch, cholangiocyte development, regeneration, and then the second is notch signaling. That'll be our second session. So we're going to have plenty of information on our website. We hope to get as many of you involved as possible. And if you have any questions about any of it, I mean, you're in this room because you care about medical research. Just come talk to me, and I'd be happy to talk to you about our event. Thanks so much.
And that's really exciting too, because get to do a symposium, a scientific symposium. The Alliance has wanted to do that for so many years, and we were so excited when we heard about this. So I think we're probably over time. I think we're at break right now. Yes.
So the 3 companies that are developing drugs, Shire's is to tackle the cholesterol problem.
No, not quite. So the Shire drug and the Alagille drug, same class of drugs, they inhibit the transporter. So what they are fundamentally doing is lowering bile acids, which is a key factor in Alagille. There are a number of potential benefits that could occur from that, and one is lowering cholesterol and having an impact on xanthomas. The others are potentially improved fat-soluble vitamin absorption, but it's really about the bile acids as opposed to the cholesterol.
The other 2, are they also aimed at lowering bile acids or something else?
Of the 3, 2 are 2 are essentially doing the same thing, and the third one is doing something different.
And that's the one you can't speak about?
Correct.
Okay. Well, I know you can't speak about how they're doing it, but you can't tell us what they're targeting, what effect they're hoping for?
No.
No, okay.
We're sworn to secrecy. Okay, let's thank Kira for her wonderful wealth of knowledge on drug development. And if you can fill out your feedback forms, we'd really appreciate it.